Okay, now I will present about the meiosis, me Nur Ayuni, Yasmin and Chuniyin. Now we will go into overview about the meiosis. Meiosis is a process with a single cell divides twice to produce 4 cells containing half the original amount of genetic information. Okay, now we will see the goals of this meiosis, which is to make the daughter cells with exactly half as many chromosomes. In human, haploid cells made in meiosis are sperms and the eggs. When the sperm and the egg joins in fertilization, the two haploid sides of the chromosome form a complete diploid, sets a new genome. Okay, now in meiosis there is 2 steps, which is meiosis 1 and meiosis 2. Now we will go into the difference between the meiosis and the mitosis. First one is the homogeneous chromosome paired at the prophase of the meiosis 1. Second, the genetic recombination, we call as a meiotic crossing over, occur at the prophase meiosis 1. Third, the chromosome number is reduced to the half after meiosis 1.
I'm Yasmin. I will present for meiosis 1.
Before we go through 4 stages in meiosis 1, I will tell a little bit about the overview for meiosis 1. For meiosis 1, the goal is to reduce the chromosome number in the daughter cell. At the end of meiosis 1, 2 daughter cells produced. In meiosis 1, crossing over and genetic combination occur. Then, there are 4 stages in meiosis 1: prophase 1, metaphase 1, anaphase 1, and telophase 1. Okay, now let me move on to the first stage in meiosis 1, which is prophase 1. The starting cell is diploid. From this short video, we can see that homologous chromosome pair up and exchange fragments in the process of crossing over. Okay, so basically this is the flow for prophase 1. First, chromosome condense and form the homologous chromosome, and then crossing over occur, and it lead to the genetic variety, and after that nuclear membrane disappear and centriole move to opposite end of the cell. Next, metaphase 1 is the second stage after prophase 1. From this video, we can see the homologous chromosome pair align at the metaphase plate. Basically, this is the flow of metaphase 1. So, the first is the spindle fiber attached to the chromosome, and after that, the chromosome line up in the center of the cell. The third stage is anaphase 1. During this stage, homologous chromosomes separate to opposite end of the cell. So basically, this is the flow for anaphase 1. First, the spindle fiber separate the homologous chromosome and then pull them to opposite pole of the cell. Telophase 1 is the last stage of the meiosis 1. Okay, from this stage, 2 new cells with a haploid are forming. So basically, this is the flow for the telophase 1. First, chromosome reach opposite end, and then the nucleus membrane form. So from this stage, we can see 2 daughter cells are forming.
So, hi guys, I'm Yi. So now let us move on to Meiosis 2. Let's jump into the overview of Meiosis 2. So the overview contains a lot of important points or concepts of Meiosis 2 that you would need to understand. So without further ado, let's jump into it. Goal of Meiosis 2 is to produce the gametes, and Meiosis 2 basically begins with 2 haploid parent cells and it divides into 4 haploid daughter cells. Meiosis 2 also is known as mitosis for haploid cells for you to understand it more clearly. And Meiosis 2 is definitely shorter and simpler than Meiosis 1 as it contains 4 stages, which are prophase 2, metaphase 2, anaphase 2, and also telophase 2. So let us begin with the first stage of Meiosis 2, which is prophase 2. So you guys can take a look on this short 10 seconds of animation on what happened in prophase 2. So basically in prophase 2, what happens is the chromosomes will first condense after Meiosis 1 and the nuclear envelope and nucleus will disintegrate. After that, what will happen is the centrosomes will move apart and the spindle fibers starts to form between the chromosomes. And the spindle microtubules begin to capture chromosomes, and there is no crossing over occurs, which means there is no exchange of information that will occur in this phase. Let us jump into metaphase 2. So enjoy a short video of 10 seconds on what happened in metaphase 2. So in metaphase 2, the spindle microtubules that connect to the kinetochore of each sister chromatid will help the sister chromosomes to align at the equatorial plane. So the one sister chromatid face each pole, so with the arms diverging. Next, let us jump into anaphase 2. And a 10-second of short animation would be better for understanding. In anaphase 2, it's very important that you remember for this stage there is simultaneous splitting of the centromere of each chromosome that will happen. And after that, the spindle fibers will shorten and cause pulling of one sister chromatid to each pole. So in this stage, it's very important to remember that the sister chromatid separate. So this is basically the main point of the Meiosis 2 that help in the cell division, which is the sister chromatid separate. Next, let's jump into the last phase in Meiosis 2, which is the telophase 2. Basically, in telophase 2, the spindle fibers will start to disintegrate and causing the chromosomes to recoil and forming chromatin. And the nuclear membranes will then start to form around each set of chromosomes and the chromosomes will decondense. Hence, the products of telophase 2 is 4 haploid daughter cells will be formed in total. And telophase 2 will mark the end of meiosis. I think that's all from us. If there is any question, feel free to ask. Thank you.